Assalamualaikum warahmatullahi wabarakatuh. Hello everyone, let me introduce myself. My name is Lintang Ayudia Nurjati. Today, we are going to be talking about one of the coolest tools in the lab, the centrifuge. So, stick around and let's explore how it works.
A centrifuge does the same thing but faster because instead of using gravity it uses centrifugal force. That's why it's called the centrifuge. Centrifugal force is the outward pull you feel when you spin around in circles. Centrifuges can spin really fast to aid the strong centrifugal force. Scientists use centrifuges to separate alkenes of particle from liquid. In this 2020 paper, scientists studied a type of fish that can change the number of red blood cells in this blood depending on whether it's resting or exercising. The researchers used a centrifuge to measure the fish's hematocrit, of the number of red blood cells in the blood. To measure hematocrit, you take a blood sample, put it in a capillary tube, and use a centrifuge to separate the red blood cells from the rest of the liquid in the blood. Scientists use centrifuges to separate more than just red blood cells. Centrifuges apply very high forces, so make sure you use a tube that's designed to be used in a centrifuge. Once you have your samples ready, you can put them in the centrifuge, but centrifuge is unbalanced if the tubes are unequally distributed, or if the tubes have very different amount of liquid in them. Putting unbalanced tubes in a centrifuge is like two people of different size spinning around. The heavier person will pull the lighter person off balance. Might be fun for people, but it can damage a centrifuge. So always make sure that each tube is balanced. If you have a tube that doesn't have a partner cross from it, use an extra tube with a similar volume as a balance. Once you've lidded the centrifuge, if it has an internal lid, put it on, close the lid, and set the speed on and time according to your protocol. The units for speed are either RPM or RCF, which is related to G-force. Higher speeds mean more centrifugal force. When the centrifuge is done, take out the tubes carefully because if you jostle them, you'll mix up the particle again. After centrifugation, the particle in your sample will be all smashed down in the bottom of the tube. The pile of smashed down particle is called the pilot, and the liquid on top is called the supernatant. When you use a microcentrifuge, make sure the hinges of your tubes are pointing out. That way you always know where in your tube to find your pilot. Depending on your research question, you might use just the pilot, just the supernatant, or both. Thank you for watching.